Okay, so this is the start of our final exam review, and we're going to start with some of the basic stuff at the beginning of the textbook that we reviewed at the beginning of the school year, but then also what we reviewed as the year progressed and we discussed it. And then this will be one video that'll be on YouTube, and then there'll be another video, other videos for the different parts of the review guide. So if I break it up by chapters, then it'll be easier for you to search certain things if you want to go back and look at specific areas. Okay, so I think in this first chapter, the reason why it's nice to come back and look at these terms, hypothesis and theory, is because when we did the evolution unit, a lot of people use an argument of, well, evolution, it's just a theory. I don't believe it because it's just a theory. But hopefully you understand that in science, a theory is something very, very different than how we use the term theory in everyday life. If you say, oh, I have a theory about that, that means kind of like I have an idea. But it's not a theory in science. It's not the same thing. So a scientific theory is something that's been developed after lots and lots of extensive support. Okay? Lots of evidence leads to a theory. And so Darwin had multiple hypotheses. When multiple hypotheses are supported, it leads to the development of a theory. The next content or subtopic that I want to make sure that you review again is dependent and independent variables. Maybe if I use like red, the writing will pop and stand out. Okay, there are other names that we can use for dependent and independent variables. Responding and manipulated. So, which variable does the experimenter have control over?
Independent.
The independent. And so we'll call this one the manipulated. The manipulated variable, and the dependent variable is the one that responds. And so when we plot them on a graph, we put the independent variable down here and the dependent variable on the side. So when it was something like human population, we saw that over time, population kind of spiked and dropped, but then in the last century, we've had an exponential growth. So the independent variable down here would be time. As the experimenter, you could look at what time of human history you want to look at. And the dependent variable was the number of people. The number of people depended upon what time period you were looking at. And then item number 6 down there just shows you a sample of a experiment, and sorry, it's hard to talk and do two things at the same time. And if I go down a little bit, designing an experiment, knowing you have a good control variable, good experimental variables, and so on. We hit this a lot in first semester, so this is going to be minimal for second semester. Okay. I'd like to spend more time, however, right now on cell reproduction and knowing the difference between mitosis and meiosis. So another name for mitosis could be just cell division. And in that cell division, you're taking one cell that is diploid, and you're making two cells that are also diploid. You should be familiar and comfortable with the terms diploid and haploid. Okay. And so if I give you the diploid number of 46, these cells down here also have 46 chromosomes. What kind of cells are produced as a result of mitosis? I heard it. Two what? Two daughter cells, exactly. So we call these daughter cells. What kind of cells in your body, however, do mitosis? Most of them, but there's a name for that. Somatic cells. Okay. Soma means body, so somatic cells refer to body cells. Okay. And I see you guys, a lot of you typing away or writing and adding to your outline. It's awesome. What are the steps of mitosis? Okay, interphase really technically isn't a step of mitosis. It's part of the cell cycle, but it's not a step of mitosis. So, prophase, metaphase, anaphase, and telophase. So PMAT, this is the actual mitosis cell cycle, and then interphase. All of this combined is what we call the cell cycle. And when we look at the lifetime of the cell, you guys have in your notebooks kind of a pie graph. And it's divided up something like that. And the vast majority of the lifetime of a cell is spent in interphase. But interphase itself is broken down. What are the stages of interphase? G1, followed by S phase, followed by G2. So we have growth, synthesis, followed by growth again. Sometimes, however, a cell will stay and not go from G1 to S. It loops back. What phase is that called? G0. So what would the result be of a cell that stays in G0? It doesn't ever divide. Examples of cells in the body that are stuck in G0? Nerve cells in the central nervous system in your brain, for example, and skeletal muscle cells are at G0. During the S phase, during synthesis, what is being synthesized? DNA, you're on fire, man. Good job, killer. One of the last times I'm going to get to call you that, too. Ah, sad. Okay, so we have growth followed by the synthesis of the DNA, followed by more growth, which is duplication of all of your organelles and overall growth in the size of the cell. Okay? Then very quickly, the cell goes through prophase, metaphase, anaphase, telophase, and divides. And you're left with two daughter cells that are both diploid, and then they're going to start into interphase over again. So that is a very, very quick review. Should you know what's happening at each step? Yes. Okay. So at each step of, if you just draw one chromosome here, okay, during interphase, this one chromosome is going to make a copy of itself. So we're going to start out, actually, here, we'll do this. We'll start out with, and I'm making the copies darker. Okay, so this organism has a diploid number of 2. Okay, because this is one chromosome and this is one chromosome. It started off see? And then they duplicated themselves. So now this is what we called a pair of homologous chromosomes. Okay? And now these two are sister chromatids. Okay, sister chromatids. Reviewing that vocabulary because it was confusing. So in prophase, you've got these guys lined up and the nucleus starts to break down. So I'm going to do a dashed line around my nucleus and the cell is still intact. Okay, that's prophase. Metaphase, shading one in, making them too big here. But I think you guys get the idea. The nucleus is completely gone, and now you've got these spindle fibers that extend across the cell. And your homologous chromosomes line up separately from each other along the midline of the cell. That's metaphase. Anaphase, they are going to move to opposite ends of the cell, and so now you still have your cell, but now you've got shaded in to represent that one, shaded in to represent that one. And they've pulled to opposite ends of the cell. That's anaphase. And telophase, you can easily envision the cell then, I should go back and just draw it on the corner here. Telophase, the cell pinches in. It's so hard to draw on this. I'm not as good as the guys on Khan Academy. They have better tools than I have. And this is telophase. So if I said here that this is one chromosome and this is 2 chromosomes, right? This is 1 and this is 2. Notice I got 1, 2, 1, 2. So now I'm going to end up with 2 cells that are each diploid. I haven't changed my chromosome number. I was able to divide my cell without changing my chromosome number because each chromatid duplicated itself. Now, how is this different from meiosis? Meiosis can be referred to as a reduction division. Because in meiosis, we start out with a cell that is 2N. But we don't keep it that way. We go through 2 cell divisions and we end up with, instead of 2 daughter cells, we end up with 4 daughter cells that are haploid. They have half the number of chromosomes. And so how does that happen? First of all, where in the body does this happen? Okay, this happens in the gonads. And the result, we don't call these daughter cells. What do we call them? We don't call these haploid cells daughter cells. What are they called? They're a special type of cell. They're not diploid. They're haploid. Gametes, I heard it, gametes. These are our egg and sperm cells. And the significance of this is that if you have an egg cell that is carrying the haploid number of chromosomes and a sperm cell that is carrying the haploid number of chromosomes, and they fertilize, the sperm fertilizes the egg, you have a zygote now that is diploid, and it has the proper number of chromosomes, and that's important. You don't want to have too many or too few. So how is it that we achieve this movement from diploid to haploid? How is it that we achieve that? What do we have to do? What? Okay, crossing over doesn't let us reduce our number of chromosomes. Crossing over is really important, but crossing over doesn't reduce our chromosome number. What are the steps for meiosis? What were the steps for mitosis? Okay, interphase, then we had prophase, metaphase, anaphase, and telophase. That's mitosis, but we've only formed 2 cells. In meiosis, we have to form 4. So what do we have to do? We have to repeat it. But what stage do we leave out? Interphase. What is the significance of leaving interphase out at this point? There is no copying of what? The chromosomes. So there is no second chromosome duplication. And this is how we go from being a diploid cell to having haploid cells. So we call these meiosis 1 and we call these meiosis 2. And they look very, very similar. Now, here's where we will hopefully clear up some vocabulary that was difficult for you guys. So I go back to my chromosomes. And I got to color them in to show my original chromosome and my duplicated chromosome, right? And then here's my other one. And its duplicated chromosome. Correct? On these 2 chromosomes, I'm carrying the dominant traits, and they both have it at this position here, okay, because they're exact copies of each other. But on this one, it's carrying the recessive gene on the tip. Now, what do you call an individual that carries 1 dominant trait and 1 recessive trait? Heterozygous. So this is a heterozygous individual. Do you see that before the duplication, it would have just been big A, little a? Before we duplicated and made that second sister chromatid. During during prophase 1, during meiosis 1, these 2 sister chromatids coil around each other and sit right on top of each other. So there's one, and then I'm going to draw the other one underneath it. It's all just, and it's so hard to draw this 2-dimensionally. I'm not that much of an artist for scribbling on it. And what do we call it? We see 4 little chromosome legs hanging out. So what's that called? A tetrad. Good. A tetrad. So what has happened is these 2 have connected together and made that. But remember, these 2 were carrying the dominant and these 2 were carrying the recessive. So since now they're all tangled up, when they go to separate out again from each other, sometimes bits swap over. They cross over. And this again is happening during prophase 1. And when they separate from each other, they crossing over can happen during prophase 1. During metaphase 1, they line up along the equator. So now you have these tetrads, 4 little bits lining up along the equator of the cell. During anaphase 1, they're going to separate. So these 2 will pull away from these 2. And this is when crossing over is going to become obvious. If during crossing over, notice the sister chromatids stay connected. But notice This one now carries the dominant trait, but this one picked up the recessive. And if you look at the other pair, this one carries the dominant trait on the end, but this one is all recessive. This is crossing over. This is just meiosis 1. See, metaphase 1 and at anaphase 1, they separate. But you keep this structure together. Telophase 1, the cell pinches in and you get 2 cells. So I gotta color this in. In meiosis 2, now these guys line up. So, you know, the cell membrane is out here. We don't have any nucleus. Imagine spindle fibers coming across the cell, coming from this side. And now in anaphase 2, these guys are going to pull apart and pull to opposite ends of the cell. And you're going to end up with one cell that gets this version of the trait, one cell that's going to get this version of the trait, one cell that's going to get this version, and one cell that's going to get this version. Do you see there all 4 of them are different? Just because of crossing over in that one little spot. So because I can't physically, pull these apart, then I can't really show you anaphase, but you can envision. Please go and watch there was that cute little video that we watched on YouTube of the square dance, the chromosome square dance. Okay, and that there's a longer version and a shorter version online, but it really does show crossing over. So now let's talk about gene linkage, and this is why every egg and every sperm is going to carry a different combination of mom and dad's DNA, even though you have the same 2 parents. But now sometimes traits are very heavily prevalent in a family. Like I have some cousins that they all have these gorgeous hazily green eyes. Just amazing eyes. They got them from their mom, and their eyes are just, you can just tell they're related through the eyes. Well, those traits, those eye traits, were inherited together, and we call those linked genes. And so the best way to talk about gene linkage is for me to show you this picture. This picture here shows you one chromosome with its copied, its duplicated sister chromatid. So first of all, you got to look at that and you got to say, okay, that is only one chromosome. But that one chromosome has copied itself. Now, as we look on the left chromosome, you see that chromosome is carrying all dominant traits, right? We're using all capital letters. And notice the one on the right is also carrying all dominant traits. Now, look at the positions of the chromosomes. The genes on this chromosome. Genes A and B are very, very close together. Can you see that gene A and E are on opposite ends of the chromosome, right? So when we look at crossing over, there is a greater likelihood that when crossing over occurs, that it's going to occur somewhere between position A and position E. There's a long distance there. There's a lot of space for splitting of the chromosomes, the wrapping up to happen. And so they have measured these in what they call map units. The larger the number of map units, the more likely there is to be crossing over in that position. So if you look at between genes D and E, 52 map units. These can be assorted independently with good reliability. Whereas only 6 map units are found between genes A and B, and so therefore those are likely to be linked genes that would be inherited together. So when this chromosome meets its homologous pair, okay, the other chromosome that looks just like it, the likelihood of when the legs of the chromosome wrap around each other, that there's going to be a break in the DNA somewhere down here, it's much more likely that it happens between D and E than between A and B, because they're so close to each other. So that's what gene linkage is. Chromosome mapping is just finding out the positions of all the traits on a chromosome. Okay? All right. Going back to our outline. So we've pretty much covered, you know, all of this stuff here on Chapter 8. Are you good? Any questions? Okay, so I'll stop the video here, and then the next part will have the next section of the outline.